And you say who you are and what you're working on. I don't know if you went to the website to look at the Google Drive we prepared before. No, maybe not. So that's what it is. So it's on the Google Drive that everybody can access. So we discussed together and spent time to try to define some question we would like to address. And then, in the next session, we have to present in less than 10 minutes what has been discussed and give some guidelines, conclusions, etc. So it's very tight. Like we have 20 minutes to prepare like 3 or 4 slides. So it's a bit, done that in the past, it's a bit complex. Okay, so Sarah already introduced during the session some projects related to development. So I won't go through what she said. And clearly there are a lot of new results related to development. And I think you will see some of the questions that we have here, they were already also briefly discussed. And I think for development, one chance we have that we often start with a question. Okay, so there's an issue, okay, why do you do single cell, blah, blah, blah. So in development, we have specific questions. So I think it's going to be something that is quite good for us. So one thing I'd like to start, because to me what is, and I've been following this for quite some time, I missed the first meeting, but I've been either going or attending online the other meetings. And it's still the same problem for development. The community is still very fragmented. And it's very hard for me to have an idea of who in the world is working on this Human Cell Atlas project and the development part of it. So I know some initiatives. So what I like to do is we won't do it today, of course, but is try to list big initiatives in the world that are related, or small initiatives that are related to human development, and try to, at the end, we don't have any, we tried to do that last 2 years ago, to have like a Google Drive where people would put their names, a list of email addresses that I could email them, contact them to build the network, and I got 3 answers. Okay, so which was okay 2 years ago. Now it's getting really problematic because I would like to have an idea of what is the community. I think it's really important also for development community to really start working together and to really provide that type of information. Because if you want, there will be, for instance, as an example, there will be a meeting in Toronto in July that will be related to the development part of the Human Cell Atlas. Okay, so it's dedicated to development in Toronto in July. But I don't even know who I should contact in the community to tell them about this meeting. So we need like to get ideas of where are the people, what are they working on, and also to try to avoid also having like 3 or 4 groups that don't know each other working on the same topic because they could work together. So I think it would be a waste of time and money, etc., if there's no coordination. So I think it's really something that's going to be important. So I'm going to have to find a way to opening an account, I don't know, WhatsApp, Twitter, something like that, to try to collect and have all of you like registering and giving us some at least names and ideas. We really have to have a list because we still don't have it 2 years later. So it's really a problem. Okay. So one thing, I wanted to give you examples of things that have been changing in the past 2 years. There was no really like development was in the middle of other projects in the past. Now there are really some projects that are really purely development. So I don't know if you're aware. So she mentioned a few, but some initiatives that are like been funded or are going to be funded hopefully. So for instance, in Sweden, there's a Swedish Developmental Cell Atlas project that exists. So this is well structured and it exists already for a few years, and I think it has been renewed. So this is one example of one country that is putting money to build this atlas. In France, so Paolo and myself, we are part of a new program that was presented in November by someone in Japan that is named Human Developmental Cell Atlas, UDK. Okay, and it regroups French groups, and the idea is not to really do a project, but it's to provide facilities and build platforms. So, for instance, with Paolo, we are building a biobank, and other people in France, a biobank of human embryo samples. For now, in Europe, we just had access to the HDBR biobank. So we are building our own biobank, so which will be a bit different. We can have like all, through Paolo, like whole embryo that are, I would say, like almost perfect, complete, which is quite rare. And we have other sources. So we have 1 year to build the database, it will a biobank, and then the idea is to open it to France and then to Europe. So I'm aware of this initiative, the HDBR, but if other countries are trying to build that type of biobank for human embryo tissue, I have no idea. So, yeah.
Maybe I could just say that within... my name is Kirsten Meyer. I'm at the Sanger Institute in Cambridge, and we work very closely together with HDBR, and also Mas Hanifa's group in Newcastle. And so HDBR is the Human Developmental Biology resource, and they have recently received quite a lot of funding from the MRC, and there's also been in England a call from the MRC for human developmental projects, and 13 different projects got funded, and there's a website where they're all listed.
Yes, in the UK it's quite well developed, and the Wellcome Trust, I mentioned the French one, so the INSERM has put 2.5 million euros, so it's not zero, to build those facilities and platforms. So the Wellcome Trust has put, I think, between 10 to 12 million pounds in an initiative that is named HDBI, and the kickoff meeting is tomorrow in London, so I'm a part of it for the imaging part, but I cannot go because I'm here. So it's only human development as well, so that's also a lot of money. So the Swedish, the French one, this one. As Sarah mentioned, there's also a lot on brain development. It's mostly coordinated by Arnold Krichstein from UCSF and also the Alain Institute, but then other people are also contributing. So, but apart from those four things that are well identified and clearly as development, I have no idea if there's something similar elsewhere in the world or in other countries. So, as I said, it would be good if some of you are aware of other initiatives like this to let us know, and those are funded. And otherwise, if you want to mention some teams that you know, or if yourself you are involved in a project that is funded that is related to this, I like to know. Otherwise, suggest something where I could try to gather this information, because it's really something that I don't have. Okay, so I don't know if someone wants to say something or contribute. Yeah.
So I'm Jorge Ferreira here from Barcelona, the CRG. So it makes a lot of sense for people to organize in countries, Sweden, France, but particularly in development, it makes a lot of sense for organ-specific efforts across countries. So perhaps that makes more sense in terms of how to aggregate data and know-how. For some organs it might even make sense to purify a certain cell type before starting to do single cell. So, I wonder what your thoughts or people's thoughts are on that.
I fully agree. I mean, there's something that we might discuss at some point, which are some specific ethical rules when it relates to human embryo. So it's a bit different. It's a bit harder to ship. So when I get samples from HDBR, I also have to get authorization from France for importing human embryo samples from the UK. So it's a bit slightly different from adult tissue. So when I'm going to send them to Germany, also they have to get local authorization. So which is not always the case. What is also different is that the samples we are getting, they come from dead individuals. So it's also not the same regulation in terms of GDPR. We heard about that. It doesn't apply to the human embryos. So in a way, it's a bit easier for some things, but a bit more difficult for others. So having different countries getting samples, it's okay. Distributing them is not so straightforward.
Sure. Perhaps I should clarify. I didn't necessarily mean shipping embryos around Europe, because there are some in every country, of course, but rather in terms of distributing efforts, because human development is extremely long. There are a lot of decisions to be taken. It doesn't make sense for one group to do absolutely everything, different interests, and so organizing and prioritizing what each group does would make a lot of sense.
I mean, I agree, but that's where we need to know who is in. So I mean, people working on kidney, I know they start on lung, they start to do part of the project is also on development, but it's only this group of people. There's a group on pancreas, and I know in France there's a group working on development of the pancreas, and here they're funding also to work on it. So, but again, I don't know where are the people, who is working on, there was a paper published recently by Seth Blackshaw on the human retina, It's on bioarchive now. I also do the same kind of thing. I didn't know he was doing it. So clearly it shows that there's no coordination. I don't know who is working on a different organ. So that's really something that is missing now. The community is not like well organized. So that's what we need to do. So I think we need to find a way to advertise it somewhere and have people like, if they're willing to, because people might be afraid of being scooped or whatever, to advertise and say they'd like to join the network, because that's really a problem.
Maybe I could just say, before the Horizon EU 2020 program, on the Human Cell Atlas webpage, we actually put out a call saying, anybody who's thinking of applying, are you happy to register here? And then maybe that can help people to coordinate their efforts for submitting samples. And the Human Cell Atlas web pages now do have a specific subsection for the Human Developmental Cell Atlas, and it would be relatively easy to, within that, set up a call. And again, just ask everybody, if you're willing to let others know what you're working on, please sign up here. I mean, we could initiate that relatively easily.
So just a few. Did you want to respond to that? So Kat Gold, I work at the Wellcome Trust. I lead on our engagement with the Human Cell Atlas. So just to maybe a couple of comments. So from the Wellcome funding for the developmental work, so there's a few strands there. So we and the MRC Core Fund, the HDBR, which is a resource that anyone can use, as long as they have their own kind of ethical and regulatory bits in place. We fund developmental work through our strategic support for the Human Cell Atlas, which is led by Sarah, but also Muzz, and that's looking at selected tissues. And then there's another strategic initiative, which is the Human Developmental Biology Initiative, which I think is very synergistic, but is not specifically an HCA initiative, and that's focused on lineage tracing. And then the other thing that I just, so Goji Go presented at the HCA General Meeting in Tokyo. So there are groups of researchers in China doing quite a lot of developmental atlasing, and I think some of the challenges there would be good to align, but data sharing can be quite challenging, I think. So it might be difficult to share data, but it would be good to develop a framework together and align. Yeah. And the other thing I wanted to mention at that Tokyo meeting, I don't know if you That's it, yes. So she presented, and Gary Bader, I think, in a breakout kind of got a group of people together to try and start thinking about the Toronto meeting. So I guess they would, yeah, the full list was kind of there. But I don't know of any other big efforts.
This meeting might help.
Yes.
There's like a dedicated meeting.
Definitely.
Yeah, for sure.
Yeah, but I agree that we need a common platform, basically. And this is more or less what we're trying to do in France as well. Like we kind of making the idea of this consortium is to have a kind of portal that is HCA and HDBR compatible, meaning that at one point the idea is to fuse all this data together with England and eventually with other initiatives, because I think this is the way to go. So I totally agree with you. And
we have a lot of pediatric researchers I work with and I know are very interested in working with people in this room and outside this room, especially if you have any interest in looking at the follow-up in developmental biology and pediatric diseases. So in Toronto, there's going to be a lot of pediatric people attending. So if you have any interest in collaborating with anyone in the U.S. at least that have a large organized pediatric community, you can ask me or Bruce Arano at Cincinnati.
At Masanifa's lab, we consume a lot of the data that obviously comes out of HDBR, and we've developed a couple of online portal platforms to consume this data and distribute like the viewership of this data across to anybody who's interested. So I thought it might be the right platform to just mention.
But what type of data? Then it's just the sequence data.
Yeah, exactly.
No.
So So the data that is that you would get on the portal would be visualizations of your data. It's a gene explorer mostly.
Gene Explorer, yeah.
Yeah, it's a gene explorer and a dimension reduction explorer for your data. Also, it comes with lineage differentiation portal and interactive heat map for exploration of any data that comes out of HDBR. It's in its infancy right now, but as time goes by, we hope to improve it. It's transcriptomics.
Because I wanted to show you, because I might not be able to, I'll try later to reopen it, but I got an email, they sent me what is available right now in terms of embryonic or fetal data on the DCP, on the portal. It's not much. Okay, so I have to connect at some point. I'll show you, but there's not much there. It's a bit limited for now. Okay, so it's really one thing to me. That's really the problem, is not to know who is doing what and where they are and if they want to work together. And I think it's a problem for like grant application, etc., because it's not very. It's not well structured. That's really a problem we have. You see the gut, the lung. No, they work together for quite some time, and it helps a lot for funding and for showing that something is interesting. So I think that should be a major thing we should try to do, because it hasn't moved much in 2 years. I mean, it's better because now at least there are some consortia that are funded and there are some clear identified projects, but it's still not the best. So I don't know if the one thing also that will be important for us, and again that's something that has to be discussed, probably not today, is also the collection of the samples. So how do we collect them? How do we stage them? Are we all agree? And when we talk together, some people talk about gestation week, other PCW, other like days. I mean, it's also, even this is not like really homogeneous between people, and then people, they make mistakes when they compare stages. So it's something stupid like this is already not decided. So we should at some point between us, we define what we want to have. That's something that is also missing. So we have our own, like, for the biobank we're building, and it's related to HDBR. I think we have the same kind of framework, because we kind of copy what you did, not to invent something else. But I'm not sure that everybody is using the same thing. And for development, that will be very important, because 1 week difference at the beginning, it's a huge difference. So I think we should also try to discuss and really say what we want to use at some point. So I don't know.
Yeah, I guess that what will be interesting to define here is more or less the availability of embryonic and fetal stages that we might have access to eventually. So we know pretty much what you guys have in England. We know what we have. Basically in France, we covered the first, let's say, from the 5th post-conception weeks to the 12th, 14th, to the max. And as Alain was saying, what we're getting in France is mainly intact embryos and fetuses. So it means that we have the full body in perfect conditions, so we can potentially dissect any organ during this first trimester of gestation. I don't know if it's there are other groups, for instance, in Sweden that can cover further stages, and even whether it is interesting for this community to go to further stages in fetal development, and if so, why? Because most of the morphogenetic processes happens during the first trimester. So if there is a reason to go, for instance, to the second trimester of gestation, we should maybe define the rationale behind it, and in order to eventually focus on the temporal time frame that we all agree with. So I don't know if you have any thought about this.
Because in the development, I mean, that was mentioned. There's also the pediatric cell atlas, which is will be like postnatal, obviously. So because, I mean, development will continue still after birth. So, but this, I don't really, I mean, there was one paper about this pediatric cell atlas that was published, but it's slightly different from this. The material is different, and I think we should at some point define where we stop. So is it like 18 years after birth, or what should be the period we want to study? So one thing that comes at some point is what is what would be, it would be nice also to know that what would be the key question, developmental question that we want to address. So just to give you an example, for the project we hope will be, should be hopefully funded by EU, the question is easy. Or you become male or female, okay? So what happens during 2 weeks or 3 weeks, between 7 to 10, when you become a male or a female, from something that looks exactly the same. So here it's something you can only address in development. If you look postnatal, it's too late. Okay, so I think it would be, and I think in this case that we something that justified to do it during embryonic development. So I think it would be important that all of you, or probably all you have already ideas around this, think about questions that are really essential, like, okay, this is a key question. It can only be addressed during development, and this is what we want to do. Plus, for the sex determination, it's nice because you compare also male and female, which most of the time people don't pay attention to in the single-cell analysis data. It's going to come, but for now it's still not the case. But in this case, there's no choice. You want to compare male and females, so also it's quite nice. So I think it will also be nice, like you think, maybe you have ideas, like, what are the key questions, or like very important questions that can only be addressed during embryonic development or fetal development, or during like adolescence, for instance. So I think that the type of thing we need to find out. I think it's more interesting than just mapping every cell type across time, across organs, etc., with no questions behind. I think it would be easier if you focus on very basic questions that are essential for development that we don't understand. So it would be nice to try to have a list of questions like this. As I said, sex determination, to me, that's one that is really essential, but I'm pretty sure there are plenty others. So I don't know what you think about this. So the way to start, like identify the questions and try to see who's willing to work around those questions with different backgrounds and different techniques, etc. So I don't know if this strategy seems the good one to you, or if you want to comment. Yeah.
Hi, I'm Alex at Biodonosti Institute. I think that's a really difficult question because, for instance, I am working in skin, in studying how wounds are repaired. And it's really well known that in mice and in human, at the first days after birth, skin can be repaired, and even huge wounds, I mean, like finger cuts and this kind of stuff can be also repaired. So it could be also interesting to study this kind of effects just also after birth, because we don't know even if internal organs can also be affected by these repair pathways. And so, I mean, that's the key question. I mean, I think it's really difficult to find this time point exactly, because you can study from 1-2 months, even after birth. So
that's exactly the question. Like, you have to explain whether during development there's a specific point where things are changing, and I want to identify what happens during this period. So that's exactly what I was saying. Yeah. So I think it's really important to identify a key question like this, because then you could have like could bring people to put together some networks and to address questions rather than just organs, which is a bit like a problem.
Yeah, I mean,
it's important, but it's not. That's not I think what we should just do. Map cells.
Yeah, I mean, I guess that the problem here is that we would need a large network of biologists who know the exact pathways or network or this kind of stuff that most computational biologists don't really know or need help from to specify those time points. I mean, because for heart it might be just 5 weeks or...
That's exactly the point.
Hi, I'm David. I work in the Simmons Lab in Oxford as part of the Wellcome Consortium. I was just going to say something similar, actually, that I know some of the first points were talking about development being development in the first few stages of organogenesis and things, but I think there is some important questions in maturation later on in development, so we look up to about 22 post-conceptual weeks, because if you look at organs like lungs, gut and things like that, there's many diseases which have errors that come later on in the development of the organ. So so there will be lots of different questions, but I guess as well as not focusing on one end,
That's exactly the point. That's exactly the type of thing I like to have, like a list of, okay, what are the key developmental events? Can we put like, something that starts from conception all the way to 18 years, and say, during this, of course, it progresses, but there are a few like major questions or important questions, and for each organ, it might not be a single organ. For the gonads, that's what we're going to do. It's also the nerves, the remodeling, or for the genitalia, etc. It's other things happening than just like a male, female in the testes or in the ovaries. So you have to take it as a complete thing. There will be also the brain at some point, the hormones, etc. So it's an interesting process. But we should like re-identify that type of things, and I think that's what people in this room and elsewhere, that's what they should do, is really like identify like what everybody will come with his own thing, but it doesn't matter. What do they think is an important developmental question that can only be addressed during development, and then can we bring like our expertise, what we will have done, for instance, for the gonads, the type of technique we're going to use can be useful as well for you to study something else in the lung. So, but I think it's really something that's the type of question we would like to have, I think.
If I may add just a few things. So, of course, we need to put together different competencies. For instance, me, I'm a neuroendocrinologist, so I work on the hypothalamus, and I have questions that are different from a developmental biologist, because for me, I'm interested in how the hypothalamus develop, and eventually how some gene expression signatures diverge in utero due to exposure of, drugs, disruptors, and disease conditions, obesity, and stuff like this. So I guess that how we want to address the questions, it also depends on the amount of information that we might get from the patients that eventually get the abortion, because this would lead the kind of analysis to different things. Like for instance, in Lille, we know that we can get informations about the BMI, the body mass index of the patient, whether she was taking medicines or not, she was alcoholic or not, and then we can segregate groups. Of course, this would be also a subject of the discussion, whether we want to define just a reference atlas. But I think it's also, I mean, as a biologist, I have some questions to pose, and for me, the most information we get from the patients, and then we can eventually correlate whether a reference atlas diverges with versus a disease condition. For instance, is the signature of the brain of a fetus that is coming from an obese mother different from a lean mother or from an alcoholic mother? So do we have some signatures that happens already during prenatal life, because we know that many diseases are prenatally programmed? So I think that these are for me, some important questions. But of course, we need to find a way to get as many information as possible from the parents, so let's say the patients that get the surgeries. So I don't know, for instance, if in Sweden you get all these informations about the...
No, that's what we said. There are some specific ethical questions that are a bit... we are working on it. Know that there are some of those large initiatives in various countries, like the one in France, the one in the UK. We're starting also, we want to include that type of data. So we're trying to go through all the administrative and ethical problems. So we'll see if we can find a way to do it. And I guess at least in Europe, it will be rather homogeneous. Other countries, it will be probably different. But in Europe, I think we have a common framework. So it should be, what we do should be, we can translate it to other countries, most likely.
So in terms of development, we cannot define yet an age, so, or...
I agree with you that the question, just doing it by organ might seem a little bit, well, that's a bit pedestrian and we can all do it. But actually, I think there's still an enormous amount of information to find. And once we do have, nice developmental trajectories, once we know which cell surface markers are being upregulated or downregulated, we can then start to predict the interactions with the other cell types. And to me, that cell-cell interactions and how that affects organogenesis is one of the key questions. And I think before we can get there, we need to do maybe some of the really sort of basic, slightly boring profiling and cataloging.
I agree. It's just in terms of if there's limited funding, I think, if it were me, I would rather find like interesting than a question and just like, okay, I work on an organ and I want to have the development because then I will have the information. That's my view. I think if there were really some, because I mean, getting funding for this is quite difficult. So it's to me as a dental biologist, it makes sense to try to identify the questions. But it's true. I mean, every single project what we saw that represented, they always include, okay, and by the way, we also do like some kind of development, and that's it. But they don't even say why they would select a specific time point, etc. It's not very clear. They get embryonic tissue, and that's it. So which is it's a bit simplistic what I say, but that's not too far. And I think it's really important for the people from the development community to be involved. There are not so many people that just. How many of you, even if we don't go through everyone, how many of you are working on like development or human development in the room? So yeah, not so many. So like not even half of the room. So and as I say, I see it from other like meetings I've been to. They're not really familiar with all this HCA thing, and because they just see it as like a single cell, etc. Which I think also not a good thing. So I think we should like also promote more this type of initiative to the development people, because they don't. I think they're missing something. so it's really a shame. They should be more present in meetings like this, and they don't. So that's why I think Toronto will be a first step, and then hopefully the next one in Paris in 2022. But I think it's really something where then we will, you really have to try to convince people that it's really important to come. So one thing that was here is that's something we discussed with Mers and Sarah. So on your perspective, would it be better to try to cover all stages? Let's say you have 3 million to put somewhere or 5 million. Is it better to cover like all stages for like a few organs, or just as something we should discuss would be maybe a better idea? You say we identify like 8 weeks, 9 weeks, whatever, and we do everything at one stage. So what would be your preference? Like try to cover everything, and even if it's not as complete as one stage, or we just do like 9 weeks and do everything, all the techniques, the proteomics, whatever, on a single stage. So, and I told her we have to do male and female, otherwise it's ridiculous. So it's at least two models.
And if one stage, why that stage?
Yeah, that's something. So but it's something we should discuss. So I say, yeah, but then male, female, 9 weeks, 8 weeks, 10 weeks, what is the best? So what would be your thought?
So I'll start by saying I don't have an answer. But I think what would be relevant to do is to look back at what happened in the days of, because these are questions that they're being recycled. And so when all the gene expression databases were being done on a, again, international level, so the same question came up, and they decided to stick with E14.5. And so it would be interesting, I think, to go back and talk to those who were involved there to see, so what's their perspective on the response after so many years? Was it smart to do just E14.5 and be a lot more comprehensive in analyzing everything, or should they have spread out and done several ages? It would be good to learn from their experience. Having said that, at that time they didn't have to do male and female.
But E14.5, the sexual differentiation already took place.
Thank you. Yes, that's what I was going to say, that this was about mouse development and about organogenesis being largely complete. I'm from the Anatomy Institute in Vienna. My name is Nele Herdiner, and we come at this from a mouse developmental perspective with a database of single mutations that knock out mice that are embryonically lethal or subviable. And here, for the phenotyping, it made sense to have organogenesis mainly completed to then compare the different knockout mice. And that's also thought of as a reference database for human diseases. Yeah.
Yeah, I was going to say something along these lines as well. I think in mouse it makes a lot of sense to select one day for E14.5, and there's a lot of things happening then. And I know personally from the standpoint of pancreas that it's really a hot day to really analyze, and probably many other organs as well. But in human, things that happen in one day happen in 4 months. So it depends on the question, as you say, depending what question you have. If you're interested in capturing progenitors, capturing specification events or maturation, but it looks like one day is like in human it might be a lot, a bet really, if you want to bet for all organs and across the board.
And that was also a bit. I mean, this question is also a bit related to some type of funding you can get. Like, as you know, in the US, working on human embryo is getting tougher and tougher. So it has since July, it's getting very difficult now. So is it easier for like a funding agency to accept that you work on a single stage, rather than, okay, I'm going to get a collection, collect tens of embryos of different stages? So if it allows us to get funding, when I say us, is that there's no community just focusing on one stage, that could be the idea. So like, so that's why we also had this thing. Maybe it would be easier for us, the people, to get funding, at least from some countries, if we just say, okay, we don't want to do everything, we just want to focus on a specific stage. But I agree it's a bit limited, but there's some reasons also behind this idea. It might be easier to be accepted if we just do one stage. So that's the reason. Yeah.
Hi, thanks. So I'm Steve Sansom. I'm at the Kennedy Institute in Oxford, and we're working on the joint mainly. And the motivation for us comes a lot from immune-mediated inflammatory disease, where, for example, you can have synovial hyperplasia, and we don't really understand how that happens. And we have the idea that if we understand where certain fibroblast populations, for example, come from in development, then we might be able to manipulate disease and to reshape the joint in the adult. So we just started to generate some data in the embryo. We've gone for one time point. And what we very quickly realized is that to answer our questions about where the different populations are coming from, we need more than one time point. We need a series, yeah.
And you're getting them other embryos through the HDBR? Or it's not
We're getting them through the HDBR, yeah. At the moment, we have kind of limited resources at the moment to do this, so we probably will stick with a single time point initially to repeat it in different individuals. But the plan going forward will be to do some kind of longitudinal, not on the same individual, obviously, but yeah.
And just to chime in, I think we're in a similar position with NeuroCrest, which differentiates asynchronously throughout development, and basically catching a couple of time points will give you a narrow view of it. So I'm sure this is the case for many organs, whatever you pick, basically.
Yeah, I mean, for me, it's
like a longitudinal study rather than a single point.
In the human ovary, females, the oogenesis and then the pruning of oocytes happens basically 20 weeks onward, so it's quite late compared to some of the early stuff.
Yeah, I mean, ideally, I do think that we should cover the whole development, but it's a matter of fundings and manpower, I guess. This is why it would be interesting to see how many groups can eventually, if we are like 100 labs doing this kind of work, we can cover the whole developmental stages. If we are like 5, I think if the idea is like to provide the reference atlas, we should start with one stage at a time and then cover it up. But of course, the interest is to cover everything until birth.
I just want to say, I think you're actually in quite a strong position as a potentially smaller community. I think sometimes it can be easier actually to come together in a cohesive effort once you've done that mapping exercise. The other thing I was wondering was how much you've talked to, so there's another HCA community which is maybe not always so well integrated, which is people working on model organisms and model systems. And obviously a huge amount of developmental biology comes not from humans. And there's a far smaller human community than there is Drosophila xenopus mouse. And I was wondering how much you were thinking about talking to them. Obviously you don't want to kind of diverge from the central point of a human atlas, but actually perhaps there will be things that could feed in.
I come from mouse development, and so I was not doing human before I met this guy. So it means that, so it means that, of course, we bring our own like ideas and ways of saying. And I do lots of mouse genetics. So in our case, we develop techniques to study mouse embryos by imaging that now we can apply to human. So last time, 2 years ago, like Scott Fraser was there, a lot of people. So it was nice. And here, I mean, this time I don't see many. As I said, it's something that is not. I think they're missing it, because you can come from a background where you do. Nicolas Douarin is in my lab, so I'm aware of neural crest cells. So it's really good to have people around like this that can give you ideas. Even they work on chick embryos, they have good ideas for human as well. So, but again, I think they will be more attracted if you say, okay, we want to address a specific question that is very interesting in terms of development, and then it might be easier for them to bring. They might just bring the ideas or techniques they develop in other species. So I think I fully agree, but they don't come, so which is a bit annoying, and I don't get it. So and there are meetings. Development as a meeting. The Journal of Development organizes a meeting I went to. It's in September this year. It was 2 years ago. I went there, and there it was a lot of development. This time it's mostly organoids. So if you look at the program, it's like 80% organoids. So which is a bit like okay, but it's not. So it's not real like development. So it's a bit different. So it's a bit. I think also that could be a way that for us to also promote what we're doing is also people who are doing organoids. They're gonna need to know what is a normal development. Otherwise, what they're gonna do is like okay, they have something that resembles something, but so that's where I think it's gonna be very helpful to have people together, like the organoid people plus the other ones, and to really work together to understand what is a normal developmental sequence. So that's why I think if they don't have the development data. Okay, they will have organoids, but I don't know what it means exactly, what they have in their dish. So I think that's really something that people should also understand. It was really, and organoids are getting very popular. They're funding around as well. So I think it's really like something that people should contribute to. So
we should move on to the next point.
There were other stuff.
Because maybe we should pass to the questions.
What was it? Yeah, so because I wanted to say something about what you said, and you said as well. So, and it was also already discussed a little bit by Sarah. So should we stick to normal development right now, or already start to think about diseases? What do you think? Because I mean, developmental disorders, it's really very. I mean, it's a big challenge. It's quite frequent, and so. And they're really like devastating. So I think it's an important issue. So is it in our case for the development, does it make sense to really start also with some diseases at some point? Should we include disease in each type of project? Or it could also be like environmental factors or endocrine perturbators, that type of stuff. So should we include this in all types of projects, or do you think it should be like for the adult organ, we should stick to normal development, normal tissue now, and later when it's known, move to something that is disease? So what is your opinion? Would you rather stick to normal and then disease, or do all of them together? Yeah.
So my view is that it's actually such a challenge in itself that we should initially just stick to normal, because I think it's also, especially when we were talking about the comparison to mouse, I mean, the human embryo is just so much larger, and it just makes it a much more logistical challenge, and I think there's still a lot to do just with normal.
Awesome. Yeah.
Not in this part of the Human Cell Atlas, but generally we found it to be a challenge to get funding if it's just documenting the normal stage. So if there's no basic research question behind it, that you compare a disease state to the normal state. just for getting funding at all.
I kind of agree. And for now, if it's related to HCA, it should be like normal. So then you're not forced to include disease, which is good. But I agree, in some countries, if it's not related to a disease, it will be very complicated for in the country itself to get funding for this. So but for HCA, for now, we can at least say that for HCA, disease is normally not so important. What I heard this afternoon was, okay, now they're switching apparently towards like a disease. So maybe things will change. But that's what I was. It might be, as you said, easier. But then it's, I don't know, we open the Pandora box in terms of groups and what is the disease then. And so, okay, so maybe more still the normal development for now. Other comments from people? No? Yeah, Anders?
Hello, I'm Anders from Copenhagen, Denmark. I'm a pediatrician and I see patients with developmental disorders, so sex developmental disorders. And obviously I think from that point of view, my point of view, the disordered situations is close to my heart, and actually understanding normal development is really the necessary part for understanding how things can go wrong and how we can potentially optimally treat it. So I would, but in terms of this HCA and collecting and biobanking, obviously differences of sex development in children is difficult to biobank, and but I would just hate the idea to exclude the pathological situations up front, even though I know this is normal development. Yeah, I think you can learn a lot from when things go wrong. Well.
You have a lot of labs on Apospadias, just Martin Poos, and they have huge cohort of individuals. And you know, they're mainly doing a lot of genetic studies. But when it comes to know what really diverges in development in these sex disorders, we don't know anything. I mean, except for some inference from mouse work or human genetics. So I totally agree. But then, I mean,
yeah.
Thank you. Raymond Wong from Center for Eye Research Australia. So I just want to add that I agree that, you know, we should focus on the normal development process right now, because there's multiple stages that we can cover, you know, so there's actually a lot of work, and I agree with what was said here. I think that the disease aspect obviously is very important, but if we can at least map the normal process, that would at least inform the stem cell community in terms of using organoids for disease model, letting them know that, you know, they are on the right side, and from their area there's a lot of, you know, disease modeling happening, so, you know, maybe that can provide some aspect for the disease knowledge in that sense. Yeah.
I'll come back with a question to you, because we'll be discussing about this later. So how do you define a normal embryo or fetus?
How do I find it? How do I define it?
How do you classify it? Based on, I mean, morphology, based on PCR, based on which are the criteria? Because this is something that we need to discuss later on. Because of course we cannot do all exome sequencing on human embryos for ethical reasons. So you always have a risk to classify as a normal fetus some fetuses that indeed they carry some genetic disorders that you cannot really spot. So how do you think you're going to classify with a pretty confidence that it's a normal individual?
Yeah, well, yeah, I'm not sure I have the answer for this, but I would imagine, you know, like if we collect sufficient
because
number.
I think that this is something that we deserve to, I mean, it deserves to talk about it, because if we want to only focus on a reference atlas, we need to be pretty sure that this is indeed a reference atlas in terms of not carrying any pathology. So it's good if we define a series of standard operating procedures, classifications, and to get as close as we can to a classification of a reference atlas. So I don't know if you, I'm imagining that. No, because you can ethically it's not allowed because you can get to the identity of the parents. So the identity of the parents it shouldn't be disclosed. Yeah, I know. Because the big problem is that you can, using this kind of high throughput technologies, you can get to the identity of the parents, right? And the parents, they sign a consentment statement, but they also sign a document in which we guarantee that they're anonymous. So there's a matter of ethical breach. So there's only some techniques that we can use. We can use some PCR for some major chromosomal abnormalities and stuff like this, but they don't really cover 100% of disease. And so it would be interesting. So in England, you don't have this kind of problem. You can do high throughput sequencing, even if it can lead to the identity. And so you don't care about.
As far as I'm aware, we can submit fetal data to the DCP. So as far as I'm aware, from England, we can submit fetal data to the DCP on the basis that the mother has given consent and because the individual we are sequencing is actually deceased.
So,
I know.
so just so to speak from the data coordination platform, at least in the UK and in many other countries, there is no concept in data privacy of relation and of genetic relatedness. So most countries, and this isn't true in all countries in Europe, a deceased individual, regardless of who their parents are, their genome can be submitted in the public domain. Now, I mean, Denmark, I think, is a very clear example. That's not true because there's like a 10-year moratorium on deceased people. But yeah, so fetal samples, if your country has that law where deceased individuals are allowed to be distributed openly, then fetal samples, like deceased adults, are openly distributable. So.
Yeah, well, so the reason why the Swedish, Mats Nilsson, Stockholm University, so the reason why we haven't released the single cell data from the Swedish developmental cell atlas project is that we learned, or Sten Linnarsson learned in Brussels that there might be some GDPR issues with doing that actually. So they strongly advise us not to upload it on the Human Cell Atlas data portal. But I think this is sort of under investigation and can be solved, as far as I understand.
Yeah. So, yeah, the Human Cell Atlas Organising Committee has worked with Wellcome and CZI and other people to establish a legal investigation into what's going on with. Actually, yeah, maybe Katrina's better to give a summary of that than me.
It's just worth emphasising that so there has been some GDPR work being done by a task force, but that's strictly for the DCP. So that doesn't apply to all the data contributors and data users who will have to work through their own individual countries' regulatory and ethical frameworks. And you and your institutions are in best position to decide what is possible with your data. So we have been focusing solely on the DCP as a central platform, but that does not negate anything that people are doing in their own labs and with their own advice. And the other thing I would say is that so Deb Henderson and Andy Kopp, who head the HDBR in the UK, I know are kind of aware. Obviously, GDPR has come in, and I think they are kind of actively thinking about this from the perspective of HDBR. So it's possible that what is possible now in the UK may not be possible in the future.
And just so, whatever the outcomes of the legal review with respects to the DCP wants to support everybody legitimately sharing this data and will make appropriate changes as required to support legitimate sharing of this data. So, yeah.
Yeah, so Mats Nilsson again. With respect to the question of normal fetuses or embryos, in the Swedish project we get samples donated from presumably healthy individuals, but we don't have no information about their health state or anything. We don't have no information about the donors. But so the way we think about it is to have a big enough N. So if we sample big enough number of tissues, any diseased tissues should stand out as being different. So if there is sort of consensus, that they tend to form the same clusters between samples, we feel more confident that those would be normal states.
I have a question. So I wanted to ask, since we were talking about the study with genetic abnormalities, which are hard to assess, what about the physical abnormalities? And kind of a side question to this is how well aligned physical development, because so first of all, sorry, I'm Zina, I'm from data coordination platform, the Human Cell Atlas. So I'm also interested from the metadata perspective. And we use Carnegie stages, and there's also the post-conception weeks. So these stages are defined based on morphological development. So how precise actually does it fit these time points from people who work with these samples, or is it a good way of defining the development?
You're totally right. What we do is to use the same criteria. So we measure, the crumb length, you measure the foot, and some other morphological criteria. Of course, we have some divergence because the growth, the fetal growth, it doesn't align completely, and it even changes from based also on whether we are white, black, Chinese. So you have different divergence and different growth. So that's, I guess, that the easiest response is what Matt was referring to. Like in France, for instance, we're getting a lot of abortion fetuses. So we get normally 1 or 2 per week. So at the end of the year, we're pretty sure of having like 80% of aligned samples. But again, that's an estimate. And this was my very question at the beginning. How can we be sure 100% that we're dealing with a real reference sample or not? Whether it is, based on morphological criteria, based on genetic criteria. Of course, I think that we don't have the clear-cut answer. Maybe it's just when we're dealing with 500 or 1,000 samples, the error limit is neglectable. But I agree with you. Even in our case, it's difficult when you stage based on Carnegie stages. You stage on Carnegie stages based on the morphological criteria, and you can diverge from this. So you can say that this is a 23 Carnegie stages, but maybe it's just a bit bigger, the embryo. And but of course there are some other criteria like the year bud formations. You need to look at the different morphological criteria. Now we've been analyzing a lot of those samples, and we have a bit of confidence in staging these samples, but it's not an easy task, and of course they're not aligned completely, not at all. Yeah.
But I'm 100% sure that with the type of analysis we're doing, we're going to be able to completely revisit this kind of the Carnegie stage. It's like old-fashioned. Okay, so it's nice, it's useful, it's okay. But I'm 100% sure that by comparing organ by organ, we're going to find like much more precise time points and events, not just like at the size or whatever. We're going to how many cells, when do they branch? We're going to see some specific markers appearing at some different point. So I'm sure that in terms of development, human development, we're going to change completely the way it's being seen. It's going to be much more precise. I think. I'm sure.
So does that mean we're better capturing post-conception weeks because we believe that Carnegie Stages will be probably redefined?
No. I think it's still okay. It's the reference and it's working and it has been around. But I think at the end, I'm pretty sure we're gonna have like better, just not morphology, but let's say this organ, that's where like SRY starts being expressed in the testis. I mean, try to find SRY expression by looking at the testis, no way. So you know it's more or less at this time point, it may vary a little bit, but at least if you have like defined like markers, a combination of markers for specific dental switch, then you'll be able to map much more precisely, and it will be, and that's where the single cell could be interesting as well. Like you will define like something that a combination or a gene or genes that appear at some point, and I'm pretty sure we're going to change the way it's being viewed. Not as like it's more like a continuum. It's not like like stages. It's a bit. It's useful for now, but at least you can compare between cases. But I think it's going to change. You wanted to say something.
Hi, I'm Carsten from Cambridge, and I'm essentially here to moderate the live chat. And I just want to forward one question from the chat that goes back slightly to an earlier topic, because there's a bit of a time delay, and is essentially how easy is it to recover samples for a given disease? Do any of the biobanks already include disease-specific samples?
So HDBR does. They have a few, not so many, because we wanted to get like Turner, etc. And there are a few, but not so many. So they have some also in Denmark. It's not so frequent, and they you have plenty if you work like at later time point, fetal development. Like I know in France there are hospitals where they do like for medical reasons they're going to do abortion at later time point. Then if you want spina bifida, if you want trisomy 21, there's no problem, to get samples. For the early one, it's a bit more tricky, because as we said, we don't really know if they have a problem or not. So we're going to have to rely on karyotyping or PCR or trying to find out. But it's a bit more complex. Unless like I got some samples because we knew the parents had some disease, and so they did like genotyping, and then we could get an embryo. But it was we got only one for this specific disease. It's quite rare. Most of the time you don't know unless the parents had something, or that 2 kids in this case that died, so they wanted to know. So, but it's not always so easy.
If you're interested in trisomies, I think that in Robert Debré in Paris they have large collections of trisomies.
Yeah, for really major, like very frequent disease, it's possible. We had like Siamese or that type of thing. It's not so frequent, but there are some. But so for some diseases it's possible, but it's not like for now it's not really collected. First of all, cost. Building this collection in France, it's very expensive. So I think also HDBR, they are well funded. We're talking of millions to build it. So if you also include like disease embryos, it's getting. I think there's not enough funding. It would be interesting at some point. It will have to be done, but no one is willing to. For now to pay for it. It's just too expensive.
I had one more comment on the stages, because for mice, for E14.5, there are already about 3 Tyler stages that cover that area, and for the morphological phenotyping, we split that into 6 stages just to get the morphological differences, and I'm not talking about any genetic differences here. And another important thing is that one part of the disease phenotype is often a developmental delay, so you would judge the embryo to be younger than it actually is by conception day or week in human cases.
This is a hop back to that earlier conversation about determining healthy state. And it's more out of curiosity, but I was wondering what opinion was on determining healthy state by clustering, because we had an earlier topic about batch correction and how it's required in every release, every press release right now, every publication. And how would you then justify a healthy state after batch correcting a sample? Because depending on the batch correction technique you use, the underlying manifold is going to be modified, and therefore, you get similar cell types clustering together. Doesn't matter if they're healthy or not. They might form a cluster anyways? And how would you then justify this in publication? Because that's what you're going to release.
So I can give you an example of what we're doing with a project with Alain in France. And the idea is that in each we're basically 3 or 4 centers. We're going to collect the gonads, and then we'll prepare the libraries, and then there will be just one center doing the transcriptomics and collecting this. Of course, this is focusing only on one organ type. To complete the response, we're also using, and especially in Alain's lab, a lot of proteomics data, and these don't really, I mean, we don't care about the batch effect when we're dealing with proteomics and 3D visualizations. And so if we match then the RNA data with the 3D proteomics or 2D proteomics, I think we can reach a consensus on that. Then I don't know if Alain wants. Go ahead.
I just wanted to, Edwin from the Alain, I just wanted to make a comment that, I mean, I think often we like to think we're completely in a new discovery phase with a new method, but actually a lot has already been done in this space in charting development and using other transcriptomic methods like RNA sequencing on bulk samples or microarrays. And that's already actually been done for brain pretty nicely. And the outcome of that was that, I mean, we like to categorize that there are stages to go across. Transcriptomically, you see a smooth progression. And this is, I think, going to do away with the stage problem. You know, it's going to become a trajectory. You map every sample to this trajectory. And already the data is there to consider what a good sampling strategy would be to capture the most of that trajectory. And so I would encourage sampling decisions to be based a little more on what's already known, because, and not on bulk, I mean, more transcriptomically. There actually is a lot known for certain organs. Because, you know, now we're just getting down to the cellular level of what that meant, but the general trends are quite known.
Yeah, totally agree.
Yeah, I'm pretty sure at the end, all those stages, they won't be used anymore. We'll be more focusing, okay, apparition of one cell type or some specific event, and not so much the Carnegie stages at the end, because it's not the way it works. It's not by sudden you have fingers growing, or like it does, but it's not that's not the point. So yeah. Well, one thing we listed is in adult organs, it's difficult to. It's possible, but not always possible to do that, like migration assays, cell migration exponent cultures. Is this something that you think should be part of like project like on this dental cell atlas? Do we want to go all the way to like doing biophysics? For example, there's a group, I don't know if you saw a French guy who published a paper in Science on how the mouse embryo develops. It was like 2 months ago. He did like measures of tension in cells, show the cavitation occurs in the embryo. Now he's doing the same thing in human. So we have information that are like biophysical properties in the embryo, the cell tension, the properties, something different from the from just the genetics or the molecular biology. So is this something that you think is interesting, or is just like too much? It's something that is also specific to development, to be able to follow migration. For the brain, people do it. I mean, you can make slices. They can look at neuronal migration. They can start testing things in slices. But so, do you think it's something that is an added value, or that's not the main point for now? Or no? No. So I think no. Okay. What? It's not particularly technically difficult. You just need people who do it, but there are plenty, like in Cambridge or elsewhere, who are doing it. It's just like and it's done a lot in other animal models, not in human. It's done a lot in mouse, Drosophila, etc. So there are a lot of people doing biophysics of the embryo and gastrulation and other things. And it's something, because here, I mean, the samples we get, you can grow organs. You can. So it's not. So you get more information. It's not an organoid as well. So you won't grow them forever. I mean, you grow them in Denmark as well a little bit. So for how long can you grow them? I don't remember.
Yeah, hi, I'm Anne Jørgensen from Copenhagen. So in our ex vivo culture model, where we culture fragments of different fetal tissues, we can culture without any negative effects at all for 2 weeks. But for some of the tissue, I mean, it depends a little bit on the initial quality. We can culture for up to 4 weeks.
And the 2 weeks, they seem to continue the development.
Yeah, during the 2 weeks of culture, we seem to have normal cell differentiation of the cells where we know a lot about how they're supposed to develop. Because of course there's a lot of uncertainty on that basic information from the beginning. But we know that for the cells where we have enough information that they seem to differentiate as expected in 2 weeks of culture.
Yeah, I think so that's one thing where we could also bring people who are doing development in other animal models to try to apply their knowledge to this, because that's also another level of, for development is very important, all the forces, how you shape the organs, how they branch, etc. Is something that is very important. You can just not understand everything by mapping like cell types. So I think that's also where we're going to need to bring some people in the field that are bringing new techniques that won't be discussed at all at this meeting here. But I think it would be also nice to have other people. Yeah.
Maybe we can also consider this in the context of going back to disease, because I think at least in our collection of fetal material, it would be quite difficult for us to be allowed to actually collect fetal samples that are with a known disease. So usually that would not be diagnosed until quite late in pregnancy, and even though we would know of these abortions being conducted, it would be difficult to recruit the women into the project, because they are usually in a bad situation, and usually the medical doctors do not feel that they are in a stage where you can, in a state where you can actually inform them about the project.
At CHOP, where I work, there's a lot of genetics counseling, and sometimes we have parents that have histories or carriers for rare diseases that end up having a fetus or with that rare disease, and the mother opts to have an abortion. I know that's difficult in the United States, but maybe in Europe the same things may happen, where people are, carrying Huntington's disease, for example. Often women often have to have an option for abortion. So there may be other rare diseases. I know in the United States we do that. There's often options.
Well, as I was saying, we can once in a while we get some pathological samples, even early one, but it's not. It's a bit complex, so to get and also to store, etc. It's not. And also sometimes they're also going to do, if it's something that they didn't know, they don't know the reason for the death of the embryo or the fetus in this case, they're going to do autopsy. So then at the end what is left for you is not very useful. So in fact what we get are samples where they know what it is. Okay, they know it's trisomy, they know it's spina bifida, they know it's a major thing. So and then you take another part of the embryo or the fetus in this case where you know that it should be more or less normal. So that's how we can get like all the brain tissue sometime, but it's is it really normal tissue or not? I'm not 100% sure. So all the late one, most of the time, if you get them, it's because there was a problem somewhere. So it's also an issue. It's also an issue.
So.
Oh, we store it? There are different ways, because it would explain you depending on the technique we do.
So we do all. It depends. So now we're doing a lot of stuff. So it depends on the stages, but we can either fix them and store them in PBS with some antimycotic agents for fetuses or embryos that we're gonna use for 3D immunocytochemistry. We can discuss about the techniques later on. So when we do 3D immunohistochemistry together with Alain, we can store the samples at 4 degrees for 1 or 2 years, and then just take them and use it.
Incubators.
Then we have fresh frozen tissues that we use for in situ hybridization. Then we have a fixed cryoprotected, sucrose embedded, and for regular immuno in 2D. We have fetus intact, or we dissect organs and we ship them. For instance, we're also part of this consortium with Pascal Barbry, and we ship over some lungs for NUCSEC. So it depends on what we're doing, and normally we try to collect either all organs in all different status, so from fresh frozen to fixed and frozen, or just kept at 4 degrees for doing whatever you want. And then of course we keep parts of the tissues for genotyping and this kind of stuff. So, but we store in all possible ways.
The idea will be like what they do at HDBR as well. You send a proposal for a project, you say what you'd like to get, and then we provide you what you want. So if you want like a fresh frozen, fresh tissue, fixed a certain way.
We don't do paraffin.
No, but so we're gonna have like, we're gonna build in the next year or two really a frozen tissue, like fresh or fixed, of all types of stages and organs, etc., are intact. But then again, there will be like a portal where you can ask for specific tissue, and then you can provide it. We will provide it. And then the idea also will be to coordinate with people in the UK, because we don't get exactly the same thing. So that's the point. What is a kind of unique is to be able to provide you a whole embryo complete with everything, and that's not something that is easy to get from everywhere. And what we're testing now in France is we're shipping samples different across the country, different ways of shipping the tissue to see which one for what we do works the best. It was done in other places, but we just want to try by ourselves to see where we can replicate things the way we distribute the samples. But pretty much we can give you, we should be able at the end to give you anything. And we're doing a pilot test with a lab at Stanford to see if we can send samples from France to the US, how easy it is. So
yeah, because now we're also trying to use this isothermal. I mean, there are some solutions that when we dissect a fresh organ, we can send it in this solution to another lab, and still the quality is good enough then to run single-cell RNA-seq if you do overnight shipment in dry ice and whatever. So we're trying to figure out which is the best way to share samples, either for NUCSEC sequencing or for single-cell RNA sequencing. We're trying different solutions and comparing the techniques to see the level of confidence in different techniques.
It was done a lot for the brain. I mean, I know Christian is getting some samples from the UK as well. So that there's a history for brain samples for sure. Other organs might not be the same, and it might be slightly different. So that's also, I think that's also important to try to, you might need to adapt depending on the organ or the technique. And that's where I think it's going to be important to have more people involved, to try to also see if what we provide is good enough that they can do something meaningful. And if you do it, if we do it like on site, and then you do it like, I don't know, in the US, you get the same results than we had with our own sample. I think that's going to be very important to make sure that you can distribute things around. So that will be, again, it's going to take, on our side, it's going to take us a year to build this collection, but you can already try to get from the UK. So
we have half an hour, we have a few points to discuss.
Yeah, because we have like 20 minutes.
Yeah, we have maybe 20 minutes.
15, because we also have to prepare the slides.
Yeah. So it depends if we want to talk about variations of individuals. Maybe we can talk about technologies and a bit of about this. I would talk about variations, the problem of variations of individuals and technologies, and then we can briefly mention ethics, since there are people that might be interesting.
Okay.
So we talked a bit about this at the beginning. So we opted as a choice to investigate gender-specific differences, so males versus females differences. I don't know if this is something that all people working on development agree, or if you don't normally segregate the two groups. So I don't know how the HDBR works, because I think it would be, I mean, at least for us, it's a meaningful information since we're working on sexual differentiation. But also, the idea is then where I was mentioning at the beginning, which kind of information we can have regarding the donors, and whether we can find a minimum information in order to limit then also some variations in the data set, because of course we can take some embryos or fetal stages, and we'll be comparing them, but of course if the mother was assuming some substances, this will diverge and have an impact on the fetuses. So I believe that it is important to get the minimum of information about the donors if we want to be quite confident on the readout of especially for transcriptomics.
Yeah, so to do with gender again, I don't know why I seem obsessed with batch correction this time around, but once again, another opinion question. We've been batch correcting by multiple variables, but it seems that gender is something that comes up quite often whenever we discuss batch correction. So do you batch correct between samples? And if so, does that destroy any differences that you would potentially see between gender? And if you don't see any differences, how do you then justify it after the fact? So in the dataset that we just published, the fetal liver dataset, we don't see any differences between gender and fetal liver. The fetal liver, yeah. And we still haven't answered this question. How do we justify, post-batch correction by sample or different variables? How do you then pull out these differences, or how do you justify not seeing these differences?
There are some organs that are more susceptible to be sexual dependent. For instance, the brain is definitely one of such organs.
Something I was wondering, so for you, are you from the beginning pay attention to male-female differences, or you don't, or you didn't?
Yeah, the Brain Initiative project for doing whole mouse brain has a split between male and female.
For the mouse, but for the human data?
For human, not yet.
Not yet. So you're mixing male and females together?
So we're doing so few specimens at the moment that it's actually not
an issue.
Not even kind of an issue. We're limited by super high quality specimens, which I'm sorry to say has been mostly male so far. But eventually, of course, we have to tackle that. Now, an interesting thing is that the findings are kind of mirroring what's been known in the field, which is there's sexual dimorphism in the hypothalamus and not really much else. That's super obvious in terms of like at least kinds of cells. There are gender signatures. So it's not, there are a few places where we kind of thought we would see a lot more than we are. Gender is one that, two hemispheres is another, where it's not easy to see differences between the two hemispheres at all, despite functional dimorphism there. So it's an important thing to do, but I'm not sure that you're going to see it everywhere. It may just be in certain places.
Just to say, there's a bigger conversation happening in biology right now, which is about experimental design, and particularly kind of gender and sex as biological variables, and a whole other slew of things. So I think it's important. Biology, I think some fields have been quite good at that. Most have been quite poor. Even people looking at cell lines generally don't look at the sex of their cells, because why would you even think of it? But actually, there may well be influences. So it is really important.
No, I mean, for what we do in this data biobank, to us, it's we really pay attention to it now, because it's going to be a major thing, not only for like sex determination, but for other things.
This could be a naive comment, but speaking of experimental design, if you go and you measure the genders and you do the batch correction together and separately, I would feel like you should be able to estimate the magnitude of the change together and separately. And if you did that, you should know if you're overcorrecting a biological variable. Does that feel reasonable?
I guess along the same lines, just technically, I think from experimental design standpoint, it can be a tough question, but once you have the data, it's not that difficult to test. So the alignment will give you some sort of a mapping, maybe incorrect, but then you can go there and actually test for some of the variables that you're worried about, let's say sex. And if they show substantial difference, then you have to worry about whether you're over-aligned or not, whether you're hidden certain things. And you know, it's also important to keep in mind that obviously there will be inter-individual variation, and you have to, whatever the biological effect you're testing for, you have to have sufficient number of samples to overcome that variation and to actually start seeing it. Again, that goes back to the experimental design question, it's tough to answer in advance because you don't necessarily know the magnitude of the effect that will be there. Yeah, but I think that type of approach can be done iteratively. And the alignment right now, it's kind of just a single pot kind of thing, but it can be modulated in a variety of ways. You can control for certain factors when you're aligning and so forth.
Okay, so because I think at the end I'd like to spend like at least 10-15 minutes because then we have to speak for like 8 minutes or something, which is and it would be good to know among all the things we said, you tell me what you think is are the bullet point that I should, or the, even if we say things around, what I should really, the message we should bring. One thing I think that when you talk about the technique, that's something also that's quite unique to what what we can do during development, as still for the embryo. I mean, we can have the whole individual in one sample, okay? So the type of 3D imaging we do, for now it's limited by the size of the, so we use actually microscopy, and we can now image like up to about 8 weeks of gestation, a whole individual, okay, a whole embryo. So it's not like you take a part, a bit of it, you have the whole thing. So which is kind of unique. For the adult, there will be time before people can do it. And now technically we hope that with some new light sheet coming out very soon, we might be able to go to maybe up to 11 weeks of gestation and have the whole sample imaged at once. So I think that's really something that is quite interesting because, and then that's where there's a need for like people. I'm starting to give samples to a company to say, okay, this is a big piece of an embryo. We have all the nuclei labeled. Can you count how many nuclei that you have in this sample? I think now, if people are developing, so we think of putting a challenge, like for people who do like software and imaging software, like where we put like a lot of data sets where it's all like the nuclear staining for either part of the embryo or the whole embryo. Can they extract the number of cells from this data set? Can they develop some algorithm to extract the whole total number of cells? If they can do it, I mean, it means that we can tell, like in the sample, how many cells you have. We can tell at different stages what is the increase in number of cells. So you can tell about cell proliferation, that type of stuff. And again, it's unique to be able to tell in an individual how many cells exactly you have. I think that's something that can be done in terms of for the embryo or the young fetus. So I think that's really a plus compared to what people do in the adult, where there's no way for now they'll be able to tell you how many cells exactly you have in a specific organ. I think for development that's something we can do because the technique allows you, the size is such that you can do it. So I think it's, but then there will be a need we're facing problems of data sets, sizes, etc. that are, I think, similar to other projects. But it's really something we'd like to be able to have people helping us, like developing, especially automatic like cell counts and that type of stuff. That'd be really useful. So if you have people around that can help us, that would be nice. So that's one aspect. I don't know what you wanted to say about the technique or.
Yeah, but I think that you can mention about the techniques if you want.
No.
Because I think that most of the focus is here, or the discussion is about these new omics techniques. And if we need to think about a project that develops over 5 or 10, 20 years, our question was to determine whether to draw a developmental cell atlas, we only want to use transcriptomics techniques, or if we want to use a combination of technologies like proteomics, MALDI, whatever. If you want to use, as we mentioned before, some specific experimental assets as we use in mouse biology, such as cell migration assays, electrophysiology, determine the properties, for instance, of neuronal activities. So all these kind of questions, it's never been discussed, and we're mainly focusing about mapping an embryo or a fetus based on the transcriptomics profiles. But I think that if we want to think in the long run, we also need to think about also dissecting some biological outcomes, such as how these neurons migrate from this area to this area, which are the forces, which is the migration that takes place, how can we visualize it using different techniques, can we monitor the electrical activities of some cells. So I don't know if you envision to combine multiple technologies in building the human cell atlas, or if it's just mapping. If it's solely like a descriptive landmarks of genes.
So we have obviously started with the transcriptomics, but one of the things we've started to do is a TAC-seq, just so that, obviously overdevelopment of looking at gene regulatory networks is very, very interesting. So having chromatin accessibility data is quite a powerful thing in terms of looking at developmental trajectories. And, the more the merrier. I mean, obviously, it'd be fantastic to also have proteomics data, and maybe if you have slices, there are now people able to do metabolomics on quite thin sections, and again, that might be the metabolic state of a cell might tell you something about how migratory they are. I don't think we know yet. So I think everybody dreams of doing lots of different things, right? But it's a question of cost and manpower, I guess.
I think that we should consider in all branches, developmental, whichever branch, that whatever we do, we must try to use a common technique. For instance, if we are using doing just single cell RNA sequencing, we should be using a technique which is somewhat being used or which is cheaper, or which is commonly used, because otherwise we will be doing thousands or developing thousands of techniques, which will lead to the common neuron, the batch effects. So, if we are using a TAC-seq, we should be using a TAC-seq by 10x, for instance, or by some company which is somewhat accessible. I think that should be the key point in the whole human cell atlas.
But I'm not sure. I don't know. Even for the brain, I'm not sure there's you have decided. Okay, this is the technique that everybody should use at this point. No, or is it a kind of? Is there one that emerged that everybody should use? I don't think so. No. No, not really, not yet. I think it's still a bit early.
Yeah, I know. Even if you want to do proteomics and this kind of stuff, there might not be a de facto technique. Yeah. But indeed, if we want to some extent to be able to merge different datasets into one dataset, we should be using the same technique.
For sure.
Because otherwise, we will be facing the same problem as we are facing all the time.
No, I agree. But for now, there's not enough around for development to be able to maybe in a few years from now, but it's hard to know which one will be the best. So
sorry, it's not an answer. I just want to say, do you think it would be possible that you could, so Ori presented a roadmap for the gut earlier on, and for example, they're not specifying yet which particular techniques, but they're saying it should be some mix of single cell or single nuclear plus a multiplexed RNA plus a multiplexed protein, because obviously you want spatial and you want transcriptomic. And sorry, we're supposed to be do you think the developmental community could get to a point where you actually have a roadmap like that based on what's been done so far? Okay.
And again, it might be different. There's much more experience for the central nervous system, but I don't know for the other organs, it's not the case yet. So there are a few groups doing something, but there's not like 10 groups worldwide comparing things. So it's going to be a bit more complex for now. Yeah.
I was just going to make the comment that I think spatial methods are really important for separating temporal and spatial gradients, with spatial gradients being very common in developmental biology. And also, in some contexts, you don't really know if you've succeeded in extracting all of the cells. I think in those situations, you really need some sort of spatial approach.
Oh, that's for sure. And as I said, I think that's also a plus. Like, when they talk about the gut, if you take a piece of gut and you compare it to a human embryo, it's there's no comparison. So for us, we have the chance that on a section or the whole embryo like us, you can have like the whole organ if you wish. So that's really an advantage over like other methods. So you can really tell how many cells, etc., proliferation rate, number of dividing cells, dying cells. So I think it's and all the techniques are quite adapted. And there's some new multiplex immunocytochemistry that are being developed, and they work fine on tiny samples, but tiny for an embryo, if it's 3 square millimeter, it's still okay for some organs. You get plenty of information from it, not for an adult tissue, but for an embryo, it seems small, but it's very useful. So I think we have an advantage over what they would do in the adult for that type of approach. Now, one thing I'd like to do is, before we conclude, is some people didn't ask things, but ask questions. But if you want to say something or discuss something, I think it's time now before we try to sum up. So if there are ideas you wanted to share or questions, or feel free to do it now. We talk about different things, but I'm sure we forgot some topics. So if you think there's a topic that we should have addressed, feel free now to say something. Otherwise, we're just going to conclude.
Sorry, just very quickly, I just wanted to put a plug in for DNA methylation, because I think it works on a very different time scales, and particularly in development, time scale is going to be a challenge. Reconstructing dynamics. And if you have, let's say, a gene turning on and turning off in development, it will be hard to reconstruct that from RNA, from accessibility, but you should be able to see traces in DNA methylation.
I totally agree.
This is a topic that has come up in other parts of the Human Cell Atlas about how do we make sure that we can reproducibly sample the same areas of an organ or an embryo even. And do you plan to combine any imaging techniques? We've already heard 3D whole mounts, but how common is that, and is that something that we should work on standardizing somehow?
The one we use is standardized. Okay, so if you want to clear any kind of organ and map cells using immunostaining, you can do it the way we do it. It will work. Is it the best method? I have no idea, but for classic immunostaining, it's perfect. So that's work. What we're trying to do now is to see, that's part of a grant, whether what we image in 3D we can reprocess to do spatial transcriptomics, or to do some other things. So can we start with our embryo and then take pieces of it and then reprocess them something else, and then try to put things together back. So that's what we want to try now. But at least if it's just in terms of mapping cells in an whole organ, doing if you have good antibodies, that's nice. What is limiting is the number of combinations you can do. So for now we can do 4, we can do multiple rounds, let's say 8, maybe 10, but we're trying to develop techniques where we can image maybe 20. But then the idea is, okay, can we reprocess the sample after to do something else? The short answer is it seems to work okay for some things, but we don't know yet for spatial transcriptomics. We know for in situs. Paolo is doing RNA-scope in 3D. It works as well. So it's okay. The limitation here is not so much the staining, is the dataset size, which is getting huge, and then we're facing problem with the images, etc., because it's very hard to handle and to share, etc. So, but in terms of technique, this is okay. There are TIFF files. So they're fine. Maybe there will be better light sheet in the future where we can do 1 micrometer instead of 3 micrometer light sheet. but I mean, you can wait. But for now, you can already map cells like this. So at least for the imaging of organs, I think the way we do it, we can image any type of cells. That's for sure. But we don't know yet if the way we do it allows you to do other types of processing afterwards. So that's what we are trained now. Maybe what we do is okay, but it's a dead end. So maybe there are other protocols around where it would allow you to extend the analysis to of your samples with other like methods afterwards, which would be nicer. But we don't know yet. Yeah.
Last question, and then we're left with like 16 minutes.
Oh, it's good. Mine's not a question. I just wanted to say, because there's a heading up there on logistics and data sharing questions around the HCA portal and data coordination. There are some people from the DCP here. I'm one of them. We've got Laura and Zena as well. If anyone here has questions about how they can get their data shared on that portal or has had problems doing that, both of those things we'd be very interested in having conversations about. So,
is there a dedicated entry for development?
It depends what you mean by that. You could probably click development.
Yeah. I think that's something that I was seeing the community is not participating. I mean, it would be nice to have like something where it's there is development, it's clear.
Well, there's not really clearly a brain section either, or a liver section. There's a facet that you can go to get the subset of the data that's for the liver. But am I understanding that the request is specifically we should be able to highlight the specific... Got it.
Yeah, I think that...
For the portal, where we are a bit something I discussed this morning. For now, for sequence data, it's okay. But if I want to put my 2 petabytes of image data, are you ready to take them? So because
if it's multiplex image data, talk to me and we'll put it in an S3 bucket.
Okay.
But no, the data coordination platform itself is not ready for the image data.
Yeah, that's one of the things. I hope tomorrow is going to be discussed because it's really getting a problem. So it costs us a lot of money to build this database. So now it's kind of okay, but we're being limited in terms of it's a limitation for sharing data. So we have them on a server for now. And then people, so they can have access to it, but it's a nightmare to download. So now we're working on a cloud system that we're going to develop, which will be easier, but it costs like 150,000 euros per year. Yep. So and it's just for like 1 petabyte, even not even. So if everybody starts putting things, we're going to need to get a huge source of funding for this.
We would really like to work with you to solve that problem.
It would be nice because it's really it's not only us, but at least now we have really facing the other project that don't. I don't think they're facing that type of issues. For us, that's really the main limitation now for people accessing the data, and also for us like to start the thing. That's the most expensive part of the process. Staining human embryos with antibodies is not so expensive, but now that we scan at higher mag, we do like 3D stitching, etc. It's you're getting like and it's really a problem now. And we don't know where to put them, so we store them in France, but we like to put them elsewhere. Because it's really, so INSERM also is thinking of developing a cloud system by itself, and they also want to try to work with us because now at least we can provide like huge data sets and see how they work. But we'll be happy, we can discuss it with something that would be important for us because it's really a problem now.
Because we can handle it for the next 2 or 3 years.
Yeah, and then so they will be stored, but it's more like the sharing and the as I said, with cloud solution. So we're also working with a company that is developing like 3D viewers. They have a nice 2D viewer, but not a 3D yet. So if it works well, then we should all even have to, be able to have a 3D viewer to go through your data sets very well. So, but maybe we also need help in terms of how do we what are the labels, what is the ontology we have to use. That's something that's going to be important because what we're going to do, if you tell us in 2 years that the ontology is not good, that's going to be a nightmare because each data set is labeled a certain way. So we like to make sure that what we do is something that seems okay for you. Otherwise, if we have to redo everything, it would be a mess. So
yeah, the image storing problem, very hard. There's lots of parallel conversations going on. There are people talking about compression.
Yeah,
There's
yeah,
a lot of conversation
absolutely.
about featurization.
So do you know if it's something that's going to be discussed tomorrow? I see there's this spatial imaging session or something.
So there's 3 different spatial sessions.
Yeah.
You could probably drag any of the 3 of them in that direction, and I'm sure you'd find willing conversationalists.
Okay. Yeah. Okay. Okay. So I think we should stop here. And one thing is, so we do we do whatever we can. But so if we discuss quickly around what we said, like more like maybe more multiple stages rather than a single stage. Maybe not the disease yet, but etc. Try to bring more of the community and advertise it better. That type of thing, that seems to be okay for you? Yes. Yeah? Okay, thank you.
Just this point about normal versus disease. The point about normal versus disease.